What's up guys, good evening my chilla party. Well, this is your NSEJS Biology Master Teacher Akshansh and welcome to the lecture number 5 for NSEJS 2025 Biology Basic to Advanced series. Now in the last 4 classes we had started learning a lot of things about biomolecules, and finally today it's a lecture in which we are going to wrap up this chapter. So in the very first class I had taught you what biomolecules are, what is cellular pool, what are metabolites, carbohydrates. Now the classification of carbohydrates. Then we learned about what are amino acids, what are proteins, nucleic acids, DNA, RNA, fatty acids, and in the last class I had taught you about the enzymes. Now today we'll continue with the enzymes. I'll teach you what are the factors that influence the enzyme activity, the classification of enzymes, and enzyme inhibition. Now, so these are the topics that the students they generally tend to skip, but I swear that these topics are worth preparing and they are very much conceptual, and you can expect a very good application based question from these topics. So without any further delay, let's continue with the class. So now we'll continue with the very first topic, that is the factors affecting enzyme activity. So there are various factors that influence the enzyme activity, but we will be dealing with the most important one. So the first factor is pH. I have taken these graphs from a very reputed research paper, so we are going to learn a lot of new things. First of all, let's talk about the very first parameter, that is pH. So please remember that the enzymes, they are highly pH sensitive. They are highly pH sensitive. Let's say they work at a very specific pH. I can give you some examples as well. Let's take the example of salivary amylase. Okay, so the first example is of salivary amylase, which works at a pH somewhere around 6.8. So we have the first example, that is salivary amylase that works around the pH 6.8. Then we can take the example of pepsin. The pH is close to 2. We can take the example of trypsin. The pH is close somewhere around 8.5. We can take one more example, that is of sucrase, pH somewhere around 4.5. So it is damn clear that the enzymes, the different types of enzymes, they will have different pH at which they show their maximum activity. So if you can look at the graph clearly states that if like all the enzymes, they have a particular pH at which they show the maximum rate of reaction, right, denoted by Vmax, that is a maximum velocity of the reaction. So that is the optimum pH, and now as the deviation from the optimum pH increases, there is a very sharp fall in the enzyme activity. So if you can see, look at the graph clearly says that at a particular pH, that is the optimum pH, the rate of the reaction, the Vmax or the maximum velocity of the reaction will be very high, it will be the highest. And as we move away from the optimum pH, maybe in the towards the, like if we are moving towards the alkaline side or towards the acidic side, the enzyme activity is going to decrease, right? The next parameter is about the temperature. So in generally the enzymes, they tend to be functional at a temperature around 25 degrees Celsius to 35 degrees Celsius, and at a very low temperature they lose their activity, they get freeze, and at a very high temperature as well, the enzymes, they tend to get denatured. Other exceptions are there, right? So we can take the example of Taq polymerase, which is a DNA heat stable or thermostable DNA polymerase, Taq polymerase, that is extracted from Thermus aquaticus, which is also used in PCR. So there are some exceptions, the enzymes that can work at a very high temperature, but generally the enzymes, they tend to get denatured at high temperature and they tend to get freezed at a lower temperature. They have an optimum temperature at which they show their maximum activity. When the temperature is below the optimum temperature, the rate of the reaction, it doubles with every 10 degree increase or rise in the temperature. Whereas once we have reached the optimum temperature, the enzyme activity will be highest, and as we go far, like as the temperature rises beyond the optimum temperature, the rate of reaction will get halved. It is going to get halved with each 10 degree rise above the optimum temperature. So please make a note of this. It is very important, and the graph clearly states the same. Now the next parameter is going to be the substrate concentration, the substrate concentration. So we can take an example of this as well. Let's say I have an enzyme which has four active sites, okay. So I have drawn a structure of an enzyme. Let's say this is my enzyme and it has four active sites. So with the increase in the substrate concentration, initially each and every active site will begin to get occupied, right? So initially at low substrate concentration, only one active site of the enzyme was occupied. Let's say as I'll start increasing the substrate concentration, the active sites, they will get occupied by the substrate, and eventually the rate of the reaction will increase. But once the enzyme gets saturated with the substrate, there will be no rise in the enzyme activity or the rate of the reaction, right? So if we can look at the graph, it says that rate of the reaction on the y-axis and the substrate concentration on the x-axis. So initially, like, there is a direct relationship between the increase in the substrate concentration and the rate of the reaction. But once the enzyme gets saturated, right, when the active site gets saturated, the slope becomes parallel to the x-axis. So the rate of the reaction at which the active site, like the enzyme, is all saturated, that is the Vmax. Okay, so at this particular point, like beyond Vmax, the rate of the reaction will not increase. Okay, so this is, here you have your Vmax. This is the Vmax. Now we have one more term, that is K. Km, Michaelis-Menten constant. So Michaelis-Menten constant, or Km value, is the substrate concentration at which the rate of the Vmax is half. Okay, so at Vmax by 2, whatever the substrate concentration will be, it will be called as the Km. Okay, so let's say if at this particular point I have Vmax by 2, right, when the rate of the reaction, when the velocity is just half, right? So at this particular concentration, the substrate level, right, the concentration of the substrate will be called as Km. Km is the Michaelis-Menten constant. Km basically denotes the efficiency of the enzyme. Km is inversely proportional to the enzyme efficiency, right? Higher the Km value, lesser the enzyme efficiency is, or if the Km value is very less, that shows that the enzyme is highly efficient. Correct? So I hope this parameter is clear to all of you. Now the next one is the enzyme concentration. So enzyme concentration, again, the rate of the reaction, it tends to increase up to a particular level, and after that the slope becomes parallel to the x-axis again. So now it is very easy to understand. Well, we can simply understand it with the concept of the limiting agent. So it is the general chemical kinetics. Up to the point at which the enzyme is the limiting agent, up to that level, there is going to be a direct proportion between the rate of the reaction and the enzyme concentration. But once, let's say you had a given quantity of the substrate, so you will keep on, you will start adding the enzyme till the point the enzyme is the substrate is available. The substrate will keep on binding to the enzyme. But once all the substrate molecules get occupied by the enzyme, after that point, whatever amount of enzyme you are going to add, it will have no impact over the rate of the reaction. Correct? So these are the four parameters that you must learn by heart. You must understand the concept. It is very important. So now we'll continue with the next topic, that is enzyme classification. Now the modern enzyme classification, the guidelines are given by the IUB, International Union for Biochemistry, 1961. Right? So the modern enzyme classification, the guidelines are given by IUB, 1961. So enzymes, they are allotted with a EC number. What they are allotted with? They have a specific EC number. Each and every enzyme will have its own EC number. So EC number, it is basically a 4-digit number separated by dots. Okay, point or full stops, I can say. So you can have like 1 dot, 2 dot, 3 dot, 1 1. Okay, so basically we have the major class. The first digit will represent one major class out of the 7 major classes, right? And then you will have the subclass, subclass, and so on, right? So what we are more concerned about, that is about the first digit, okay, because it will tell us about the major class to which the enzyme is belonging to. Correct? So basically how the number can look like. So here you can have 1 dot x dot x dot x. Now we are not concerned about the subclasses, right? The xxx we are concerned about, we are more interested about the very first digit that will represent the major class of the enzyme. Okay, so there are 7 major classes for the enzyme. I'll write about it, and also I will tell you what the classes tell us about. Okay, so EC number 1, 2, 3, 4, 5, 6, 7. Okay, so here you have the EC. Now you will have to remember the sequence. Okay, 1, 2, 3, 4, 5, 6, 7. You cannot mess up with the number. Correct. So now the first is of the oxidoreductases. Oxidoreductases. The first one is oxidoreductases. Now the next one are the transferases. The second is transferases. The third is the hydrolases. So now the fourth one are the lyases. Then we have the fifth one, the isomerases. Then the sixth one are the ligases. And the seventh EC number shows about the translocases. Okay, so now I will tell you about each and every single category. So let's move on to the first one, that is the oxidoreductases. So now what are oxidoreductases? They are the enzymes that are involved in the reduction oxidation reactions, right? So reduction oxidation reactions, if I have to put a general formula for it, so let's say I have a substrate which is S, it is in the reduced form, and I have another reactant S dash that is in the oxidized form. So how the redox reaction will look like? So let's say S reduced will get oxidized, and S test that was present in this oxidized form, it will get reduced. Okay, so S oxidized plus S dash reduced. So this is about the oxidoreductases. Some examples I will tell you about the examples, don't worry. Okay, so now we can move on to the next category, transferases. So the transferases, they transfer the functional groups. They carry out the reactions in which there is the transfer of the groups. Correct. So one example, how, like if I have to write a general example, let's say I had the substrate S, it was having some group G. I have another reactant that is H dash. So now what will happen? The enzyme transferases, what they will catalyze? They will catalyze the transfer of the G group to H dash, right? So you will get S dash G, the group has got transferred, and you have got S. Okay, so this is about the transferases. Now hydrolases. So these are the enzymes that carry out the process of hydrolysis. Hydrolysis of what? Ester bond, ether bond, peptide bond, glycosidic bond, right? We have learned about these bonds, right? So let's say lactose. So lactose, it has a glycosidic linkage between a galactose and a glucose subunit, right? So if I'll treat it with lactase, right, which is in hydrolase, so what will it do? It will break down this lactose molecule into glucose plus galactose subunits. Correct. So hydrolases, they do? So basically, they hydrolyze. They causes the hydrolysis. They cause hydrolysis. Hydrolysis of what? Of ester bonds, or maybe we can say ether bonds, or maybe peptide bonds. Peptide bonds are amide bonds only. Okay, and then glycosidic bond, etc. So now the next category, that is lysis. So they also break down the bonds. They also cause cleavage of the bonds, removal of the groups, but without hydrolysis. And also there will be this establishment of a double bond. Let's say lysis. So as I said, there will be no involvement of hydrolysis. There will be no hydrolysis, but still there will be bond cleavage, groups removal, and also there is going to be the establishment of double bond between carbon-carbon atom. Let's say I have, okay, if I have to write a formula about it, so let's say this is what I had, C, and So lyase enzyme, what will it do? It will remove the XY group and it will establish a double bond between C and C. Okay, so you will get a double bond here and X and Y will get removed. So here you have the lyases. Now isomerases. So isomerases, they will cause the isomerism. So let's take the example of phosphoisomerase. What does it do? So we know glucose and fructose, they are functional group isomers. Okay, so it converts glucose 6-phosphate to fructose 6-phosphate. Correct? So isomerase. So isomerase, I hope it is clear. They cause the isomerism. So isomerases, what they do, they cause the rearrangement of the atom simply, right? They generate isomers. Then we have the ligases. So now the next, that is the ligases. Ligases, they cause the bonding of the chemicals. They cause the bonding of the chemicals. Now we have to establish a new bond. So we'll have to supply it with some energy. So the energy comes from ATP hydrolysis. So there is going to be ATP hydrolysis. Energy will get generated. Now that generated energy will be utilized to establish a new bond. The new bond, it can be established in the form of a carbon-carbon bond, carbon-sulfur bond, carbon-oxygen bond, or a carbon-nitrogen bond. Okay, so here we have the category of ligases. Now then the last category, that is of the translocases. So translocases, basically what do they do? So translocases, they cause the movement of maybe some ions or maybe some organic compound across the membrane. Okay, translocases, so they are involved in the movement of the ions or some organic compounds or some other compounds or some other substance across the membrane. So translocases. So don't worry, I have again I have brought a table about the enzyme classification from a very good research paper. Okay, so let's say the first category we can talk about, that is the oxidoreductases. If you can read, EC1, oxidation reaction involves the transfer of electron from one molecule to another. So oxidoreductases basically carry out oxidation reduction reactions. Example, lipoxidases, dehydrogenases, glucose oxidase, right? Then we have the transferases. So they catalyze the transfer of group of atom from one molecule to another, right? You have aminotransferases, transaminases, then hydrolysis. So hydrolysis reaction involves the cleavage of substrate by water. Example include lactase, proteases, trypsin. Then we have lyases. So catalyze the addition of groups to double bond or the formation of double bond via removal of groups. Okay. So example में pectase, lyase, decarboxylase, hydratase. Okay, then we have the isomerase. So catalyze the transfer of groups from one position to another of the same molecule. So right, it will cause the isomers. It will generate isomers only. Topoisomerases, glucose isomerases, right? Then we have the ligases. So they catalyze the joining of two molecules to form a new bond, right? You have the glutathione synthetase, aminoacyl tRNA synthetase, and then we have the translases. So catalyze the movement of ions or molecules across membrane or their separation within the membranes, right? Ubiquitin reductase, ATP synthase, ascorbate, ferri reductase, etc., etc. So here you have the examples as well. I found this table very informative. I hope at least you don't have to remember all the examples, but you should know about the definition, right? The general functioning of the enzyme. Okay, now we can move to the last topic, that is about of the enzyme inhibition. So I'll make a flow chart for you first. So we are talking about the enzyme inhibitors. So enzyme inhibition, it can be a reversible inhibition, or it can be an irreversible inhibition, or it can be the allosteric inhibition. Okay, so we are talking about the types of enzyme inhibition. Enzyme inhibition, right? So you can have reversible, irreversible, or allosteric enzyme inhibition. Now they further have more categories under them, right? So for reversible inhibition, you can have competitive, non-competitive, uncompetitive inhibition. Competitive, non-competitive, uncompetitive, and even if you refer to some very good books, there would be given mixed inhibition. Okay, mixed reversible inhibition. For irreversible inhibition, so it is group specific inhibition or it can be suicide inhibition. Okay, so this is about the whole enzyme inhibition classification. Now you don't have to learn about each and every single category. Well, in NCERT only competitive inhibition is given, so I will teach you about competitive inhibition only. Well, if you are very much interested into this topic, you can read about other types of inhibition from some good books or maybe from some research papers or through internet, right? They are very interesting to learn about. So it is very interesting to know about these types of inhibition, very much conceptual. So since NCERT only mentions about competitive inhibition, so I'll explain you about competitive inhibition only. For other types of inhibition, even if you'll refer to the books up to the level of NEET or maybe above NEET, you are not going to find all these details, right? These topics are taught at the level of graduation or maybe at post graduation. Okay, so I have just told you about the names. I will be only discussing about the competitive inhibition. Okay, so let me just draw the lines. Correct. Okay, so done with this, let's start with the competitive inhibition. So what is competitive inhibition? So now the inhibitor. What is an inhibitor? So inhibitor is going to be the substance that is going to inhibit the enzyme activity, right? It is going to suppress the enzyme activity. So in competitive inhibition, what happens? You have a substrate, you have an inhibitor, which is very similar in its structure to the substrate of the enzyme. Let's say I have this enzyme. I have this is my enzyme, correct? And here the blue color represents the original substrate. Okay, so this is my enzyme and this is my substrate. Okay, so now you will have an inhibitor. It is going to be very much similar in its structure with the substrate. Okay, so let's say what can I do? I can change the color. Okay, and here is my inhibitor. So the inhibitor, it is very much similar in its structure to the substrate, obviously. So what will happen, since it shares the geometry, and in the last class I had taught you about the induced fit theory, right, which talks about the flexibility in the enzyme specificity, correct? So what can actually happen, though, since the inhibitor, it shares similarity with the structure of the substrate, so it will also have a tendency to go and bind to the enzyme. Okay, so the inhibitor, it is also called as substrate analog, since it shares similarity with the structure of the substrate, so it can be also called as substrate analog, correct? It can also be called as a substrate analog. It is going to compete with the substrate to bind to the enzyme. Why? Because it shares, like, it shows similarity in its structure with the substrate, correct? So we can take an example of competitive inhibition, that is malonate, that competes with succinate to bind with the enzyme succinate dehydrogenase. Okay, so I have this enzyme succinate dehydrogenase. So its substrate is actually succinate, right? The name itself suggests us succinate is the substrate. It is the substrate, but the inhibitor is going to be malonate. Okay, so the malonate is the inhibitor. So if we go back to the Michaelis-Menten graph, right? If we go back to the relationship between the rate of the reaction and the substrate constant, what will happen? So this was the graph, right? Here you have the rate of the reaction, and here is the substrate concentration. So the graph was looking something like this. Okay, I'll just correct this graph. Correct. So the graph was looking something like this, and this was the Vmax, right? This was the Vmax, correct. And here we had the Vmax by 2. Corresponding value of the substrate is going to be the Km, right? This was the Vmax by 2. Okay, so now how can we overcome the competitive inhibition? We can simply add more and more amount of substrate. So if the system is going to be saturated with the substrate, you will have more amount of substrate, less amount of inhibitor, right? So the probability of substrate binding to the enzyme will increase. That's how we can overcome the competitive inhibition, right? So in the presence of inhibitor, there's going to be change in the Km value, right? So basically the graph will shift like this, right? So this, the red slope is in the presence of competitive inhibitor. So I can give you a quick summary about enzyme inhibition. So let's go back. Enzyme inhibition, it is classified into three major categories, right? You can have reversible inhibition, irreversible inhibition, allosteric inhibition. Reversible inhibition, competitive, non-competitive, uncompetitive, and mixed. Irreversible, group specific inhibition or suicide inhibition. Then we have the allosteric inhibition. So in NCERT only competitive inhibition is given. What is competitive inhibition? So competitive inhibition basically will have the inhibitor which is going to have very similar structure to that of the substrate of a particular enzyme. Since it shares the similarity in its structure with the substrate, so what will happen? It will start competing with the substrate to bind to the enzyme, right? One example we have is of the succinate dehydrogenase. So the substrate is in succinate, but when we add malonate, so malonate acts as the inhibitor. Why? Because it has a very similar structure to that of substrate, that is succinate. Okay, so how can we overcome competitive inhibition? So we can simply overcome the competitive inhibition by increasing the concentration of the substrate, right? So now how? So if the system is going to be like, if the mixture is going to be saturated with the substrate, the probability of the substrate binding to the enzyme with respect to the inhibitor, right, the probability will increase. So that's how we can overcome competitive inhibitor. So there is going to be a forward shift in the graph, in the Michaelis-Menten graph, right? The Km value will increase in the presence of competitive inhibitor. So as I taught you that the Km value basically it is inversely proportional with the enzyme efficiency. So an increase in the presence of competitive inhibitor, the efficiency of the enzyme decreases. Why? Because the Km value increases. So that's all from my side, guys. Good night. Thank you. I hope you got to learn a lot of new things. Well, if you have any doubt, you can mention them in the comment section. Else, we are done. We'll meet in the next class with a new chapter. Thank you very much. Good night. Take care.